by the Temporal Genomics Working Group, which is one of the working groups in the Research Coordination Network for Evolution in Change Disease, or the RCN. So on the chat, I'm going to be posting links for our working group. If you want to learn more about our working group or the RCN, there are other working groups. I'm going to post some links to those in the chat after I'm done speaking. So our working group is very interested in highlighting the best practices and new methods for doing temporal genomics work. So that's our main focus. We're also running this seminar series. So this today is our 5th seminar out of 8. So we actually have 3 more coming. So we really hope that you guys can join us on the next 3. So those do include topics, emerging topics in temporal genomics, as well as detecting selection with temporal data. So we really hope that you guys can make it on the next few seminars as well. Please note that we don't have time in this series to highlight all the amazing work that people are doing with temporal genomics. So to offer an opportunity for even more people to present, especially could be any undergrads, grad students and postdocs. We are running a Twitter conference on June 7, from June 7 to the 9th. If you don't know what a Twitter conference is, don't worry, you're not the only one. A bunch of us are in the same place. But we do have a lot of information in our website that can give you a head start and give you a sense of what you need to do to prepare yourself, what to expect. So all of that is in our website as well. And if you want to participate, I'm going to include a link in the chat. We are accepting abstracts right now, so you can submit an abstract now or take your time. This link will be open until June 7. So all of that is going to be in the chat soon. So we are running a traditional Zoom style seminar. So we ask you to please remain respectful, remain muted while the presentation is ongoing, and to hold your questions until the end of the presentation. Once that is over, then either Katrina or I will call your name and you can ask a question face to face. So we ask you to either use the raise hand button or just type in the chat that you want to ask a question. Then you can unmute yourself and ask a question to David. So today, I'm pretty excited to introduce Dr. David Jacobs. He's a professor at UCLA. He's our guest speaker for today. So David did his undergrad in Cornell University, he did his master's in the University of Chicago. He did his PhD at Virginia Tech. And before he started his extended career in UCLA, he did a few postdocs in Berkeley and the American Museum of Natural History, where he led a project, NASA project, that was using genetics to investigate Cambrian radiation. So since I came across David's research, there was 2 things that I always remember and got stuck in my mind. And so the first one is that his research already had a temporal component to it. At the time, I was not used to seeing a lot of temporal scale in a lot of the work that I was reading. So he already had that component, and often bringing a historical context to diversity. And the second thing is just the diversity of the approaches that David was using to answer these questions. So he studied the fossil record, large-scale physical processes, and also molecular and morphological data. He's also worked in a range of systems, including both habitats and organisms, from the deep sea to all the way to coastal, including estuaries, lagoons, and things like that, and also a range of animals and organisms, from basal animals like jellyfish, all the way to cephalopods, fish, birds, and also work with cryptid species, endangered species, extinct and living species as well. And naturally, a lot of his work is being done in California, but also in Baja California and the Sea of Cortez, which is like a special place in my heart. So with that, I'm pretty excited to introduce David for today's talk. And his title is Putting Observations from Ancient DNA and Coastal Genetics Studies in Context. anthropogenic extinctions of dikes and rivers, and coastal connectivity controlled by glaciers and rainfall events. David, the room is yours.
Cool. I just started my slides. Hopefully people can see them.
You might need to share your screen still.
I always do that.
Yeah, same here.
Sorry about that.
You're all good. Perfect.
Okay, so yeah, I'm sort of a geologist or paleontologist who's gone astray. So I study the basically both the evolution of Earth and the evolution of organisms on it in some variety of combinations. And so I'm going to tell you today about putting observations from a variety of data sources of ancient and modern DNA in context. And so we'll talk about anthropogenic extinction of ducks and rivers and some different scales of coastal connectivity. And without further ado, let's talk about two dead ducks. So we'll talk about evolution, ecology, possible future extinction of these two ducks. One of them on the left, and we don't really know what this looked like because we don't know it from the bones from archaeological sites, is a large flightless duck that lived on the California coast, but went extinct because people ate it about 2,400 years ago. The last ones were eaten. On the right, we have another duck, the Labrador duck, relatively well-known duck that basically hung around on the east coast of the United States and got shot. So Audubon painted it, but it went extinct in the 19th century. And little is known about its breeding range, but it wintered, sort of in the New York area, and that's and met its demise there. So starting with the West Coast duck, these are from middens or the garbage dump features of past folks who lived on this coast for the last 10,000 years or so. And there are a wide distribution of sites where these flightless ducks are found. So what we wanted to know is, is this, as people had suspected, a sea duck, evolutionarily, ecologically, and otherwise? And so for this purpose, let's just talk about kind of a very crude idea of what ducks are. There are three sort of general kinds. They're dabbling ducks that stick their heads down. They're diving ducks that go underwater and swim around some. And then there's sea ducks, which are deep divers and are rather exclusively eating invertebrates. So these are basically kind of also in a gradation from dabbling ducks being vegetarian and sea ducks being, carnivores or invertebrates. So we got a variety of material from museum collections and from these middens, extracted them using standard ancient DNA practices, and used a full mitochondrial duck genomes and goose genomes as bait. And so this means that you basically, biotinylate the RNAs that are copies of all that material, and then you can pull that out with magnetic beads associated with streptavidin and get just the DNA of interest, or primarily the DNA of interest, or high grade the DNA of interest from a degraded sample. And so doing that, we got a bunch of data. And so we were able to actually have success with the museum specimens and these archaeological material from the Candites, this large flightless duck. And what we found is that Candites is not a sea duck evolutionarily. It's actually sister to dabbling ducks. So that's pretty interesting. So we have this lineage that is doing something we didn't expect. We can then go and date the tree we generated with these data and all of the other mitochondrial data from ducks, which there's a lot of, and using various fossils. And we come to the conclusion that basically Candites separated from the dabbling ducks about 17 million years ago. So What we can then also conclude is that folks got to California and they ate an ancient relictual lineage, and that evolutionarily Candides is not a diving duck, but is it ecologically a diving duck? And so we can think about this in the context of this big kind of view people have of the West Coast food chain where kelp and sea otters are important. But maybe there were other interactors. Maybe this duck was here also eating invertebrates. And so that's something we want to think about as well. So what do we do? Well, we do what other folks have traditionally done in this context, looked at nitrogen and carbon isotopes from the bones and from modern material and compared them. This is comparing in red are the Candides samples. And what we can see is that there's this gradation from more vegetarian dabbling ducks to less vegetarian diving ducks to even less vegetarian or more higher trophic level sea ducks, and then we get up here to Candides. So we can come up with a conclusion that Candides is like beyond, it's an uber sea duck. It's beyond other sea ducks, if you will, even though it's not even a sea duck evolutionarily, it's even more so ecologically. And so we can now ask questions about in the past with trophic interactions with these other players in this system. So here we took some other generated comparable information from sea otters, from harbor seals, and from Brandt's cormorant, and to just sort of see what might have been going on with interactions between these. And what we then find is that basically Candides is overlapping significantly with otters, not at all really with Brandt's cormorant, maybe a little bit with harbor seals. And so we can then come to do this kind of analysis where you try to make inferences through a time series about the relationships of all the players derived from archaeological material. And so what we can see is through time, ducks declined, but oddly, if you will, otters went up. We think of otters as having met their demise with people, but initially people and otters got along, or as the human population increased, the ducks went down and the otters went up, and other things happened, like abalone got smaller, perhaps as otters and humans were better able at taking larger abalone than the ducks were. So the implication is that there was competition between the ducks and otters, and the elimination of the ducks ultimately by people led to otters being able to expand. And this is obviously prior to Western impacts on these systems. So we just published this work with my primarily driven by Terry Jones, an archaeologist. So we can sort of think of this in a broader context. When folks got to the West Coast, they probably ate things like this first, the stellar sea cow, which was in California earlier in the Holocene, and is now extinct. Subsequently, you eat smaller prey, perhaps, and so we have this implication of what's going on here, why we might have the late elimination of sea ducks, and that there were other trophic players in this system trophically in the past. It wasn't just otters and kelp. You had a more complicated system with other invertebrates, especially this particular flightless duck. All right, so let's move on to our other duck. It turns out the Labrador duck and all the other ducks we got were all sea ducks, because we thought we were going to find that Candides was a sea duck. But it turns out the Labrador duck is, unlike Candides, it is a sea duck. And what's interesting about it is that it's actually sister to a modern duck, Steller's eider. And Steller's eider is actually the rarest of the Northern Hemisphere sea ducks. And so that's sort of interesting. We have an extinct duck and a rare duck being sister taxa, and they actually diverge at the same time as the cooling of the Northern Hemisphere, the dramatic cooling. And this makes a little bit of sense, because if we look and try to inform ourselves about the breeding ecology of Labrador ducks from Steller's eider, we may be able to get somewhere, because Steller's eider basically only breed in these pools that are actually on frozen ground. These are permafrost dependent breeders, and which is one reason perhaps why they're getting rarer. The ranges are retreating as the permafrost melts. And so these are grassy ponds that are perched up on frozen ground. So we tried to do with an undergraduate, Cami Pollock and some other colleagues, we were doing some maximum entropy modeling of the range of Stellar's eider to better understand what was going on. And what's intriguing about this is Stellar's eider nests in Alaska and northern, basically northern Eurasia, in areas that were actually ice-free in the last ice age. That is, they weren't glaciated. What's also interesting is there's this area over here which is not inhabited by Stellar's eider, and we posit that this may have been the breeding range of Labrador duck. And so this is this model is based on a variety of factors, and the implication is then that the Labrador duck were over here, and the Stellar's eider are northern Eurasian taxon, and that the evolution of permafrost with glacial cycles has something to do with this isolation, and also how these ducks were probably behaving during full glacial advance. So that's a pretty interesting way to hopefully we'll get this published, and it will give us some insight, or people will be convinced we have some insight that we didn't have before about the breeding habitat of this now extinct duck. And so this is what we know about it. Here's where they really all were shot. And people have speculated about where they might have been otherwise, but there's no actual breeding data from breeding known. And so this inference that they were up in the Fox Basin here is where we, the conclusion the inference we come to. All right, so let's move on to some extinct rivers. I'm not sure if that term is most generally used, but I'm going to look at two examples of sort of extinct river problems. One is in Southern California. Turns out people don't like having their landscape flooded, and so people wound up taking over this landscape of the Los Angeles Basin, which is basically an anastomosing set of rivers and streams that flood intermittently. Since I've been in LA, which is about 25 years, it's rained less than 4 inches in a year and more than 40 inches in a year, which is nothing like New Jersey, or I understand you're from other places as well, but the discontinuity of precipitation, or the variability of precipitation is ridiculous. And the behaviors on the landscape mean that, sometimes the whole LA Basin would be effectively flooded. Most of the time it was dry. So in the early part of the 20th century, people got rid of these systems and made them into ditches, concrete lined ditches, which I guess are famous for being historically for being skateboarding locales, but I don't think anybody skateboards in them anymore. Anyway, they no longer supported their freshwater biota, so they are effectively extinct rivers. And so we were interested in the organisms that existed in these freshwater systems, and one taxon is Syncharis pasadinae, which is a now extinct shrimp, which used to live in Southern California. Its congener, its only other close relative, is up in Marin County, effectively in Northern California. Well, there also were some unionid bivalves. As people know, unionids are often endangered in various systems, and they're locally differentiated in the West in different river systems. So we might speculate that the unionids in the LA Basin would have been distinct. And we're also going to be talking about in more detail unarmored stickleback, which are an endangered taxon that still persists near the LA Basin, but used to live in the LA Basin. And so we'll talk about that. And so these are, so all this work is based on museum collections. That is, we're getting material from museums, material that, normally people hesitate to use because a lot of it is, in this case, are formalin preserved, but we are doing various things with these taxa because each of them presents a different question, right? We're trying to figure out, something about the sister taxon. Maybe we can reintroduce it. So we're sequencing hopefully the sister taxon and this ancient shrimp, or this extinct shrimp from the freshwater shrimp. Here we're using a technique very similar to or approach we're in the process of doing an approach that used the same mitochondrial bait approach we'd use on the ducks. But we're all doing whole genome resequencing on the unarmored stickleback because we're already doing whole genome resequencing on other samples of unarmored stickleback. So I'm going to mainly talk about this and not the others from here forward. But this is an interesting situation, so you can get a general idea where these samples are from in the LA basin from the museum specimens. And we can just look at what happens when you try to get data from these taxa. The clams are not formalin fixed. The shrimp and fish presumably were fixed in different ways. But depending on the sample you look at, you sometimes wind up being lucky. And you do these extraction protocols. They're actually not particularly novel or interesting, but in some cases you recover significant amounts of endogenous DNA as opposed to bacterial DNA or other contaminants. And so some of these samples like this clam and this fish or possibly this shrimp are really seem to be good choices for this kind of interrogation. And so what we're doing now is actually looking at some of those stickleback in more detail because we're already working on a resequencing project for what are the unarmored sticklebacks. These are distinct from the low plate stickleback. So in the northern climes, once the ice age is retreated, stickleback got into fresh water where they evolved loss of this armor associated with marine forms and became low plate. But in Southern California, there's a number of populations that actually have no plates. They're completely unarmored. And they are also in a situation that was not glaciated. So they might be more ancient, they might be interesting, and also they're listed, one of them is listed as endangered taxon, federally listed and state listed. So if we look across Southern California, here's where stickleback remain today for various reasons. And what we find is that here's the type locality of this Williamsoni unarmored form. But it turns out that unarmored fish are also found at these other locations. So this is sort of interesting. They're all upstream locations. In this case, they're on the top of the San Bernardino Mountains. But this is a very complicated situation, and the U.S. Fish and Wildlife Service is interested in trying to understand this better to better preserve this endangered taxon. And I can show you some of the complexity. First of all, folks were busy moving fish around accidentally with trout as they move trout around. And these are places that might possibly have been affected in that fashion. And another aspect of this landscape is basically it burns down all the time. You might have heard about this in the news, but basically all the highland areas have burned at least once. These are all just a map of fire scars superimposed on the stickleback sample. So basically most of the natural stickleback populations have been affected by fire. This is a fire that was burning a few days ago, actually. So we can see that this is a landscape that's affected this way. And of course, the streams don't do that well when the landscape around them burns down. So there's a lot of sediment load and often fish are lost from the streams. So management has been busy over the last 30, 40 years, 50 years, moving fish around from the type locality to other refugial localities. And so there's been a number of movements that have taken place to try to preclude the elimination of the endangered taxon via fire. So you've got a complicated problem that you might want to understand in terms of what's going on with these fish. So here I'm just showing you a principal components analysis of 72 fish that were whole genome resequenced by our group, my student Rachel Turba and others. And we can see that there are various interesting features here. And I'll go to a slightly things spread out in intriguing ways. And we can see basically what this means. We have previous microsatellite data that we looked at this with. And what we can see from this is that we have these places on top of the mountains where there are unarmored fish. We have and outer coast fish where one stream upstream has unarmored fish. We have the type locality down here. So basically we have unarmored fish in 3 different genetic backgrounds. And so that's pretty interesting. And we also have some of these places that are intermediates, which seem to be places where there were introductions via trout. And so there's a number of interesting implications of the data we're getting from the whole genome sequencing. But getting back to the question we started with, we're interested in getting more data from this and other fish from the LA basin, from museums, to figure out what happened in these extinct rivers. Which of these taxa what taxa were in here that are related? You know, perhaps which taxa should we try to restore? Or maybe I shouldn't say taxa, but which of these genetic elements should we try to restore to the now eliminated rivers of the LA basin? I have a lot of these rivers are not plotted because they're not, you know, we didn't get fish from them. So that is perhaps one way to put this question, if you will, that we're trying to address here. So again, using ancient fish or museum material, if you want to call that ancient or not, I suppose is a matter of choice, to try to figure out what was in this now sort of eliminated landscape. So that's what we've been doing, and much of this work has been done by Rachel Turva, and I think we're going to make certainly a lot of progress with the stickleback, hopefully with the clams, and maybe with the shrimp. We'll see. Another situation in nearby, or not right at LA, but related to LA, is the Colorado River. And one of the reasons it's related is because we in the US take all the Colorado River water out of the river, and we'll get to that. So this area here in the upper Gulf of California is where the Colorado River, a very large river system, enters the sea. We can see that in this region, and we're looking down the Gulf in this shot. This is the 1,200 kilometer long Baja California Peninsula, a very long, narrow seaway formed tectonically. And we're interested in this particular case in this region here, which is the historic delta of the Colorado. And we can see the US-Mexico line here just by the difference in irrigation practice. But this is the delta region. And so if we go to the delta, one of the things that we found is that there's this, I went down there and I was catching fish in these deep channels. It's cool to work here. The tide is 10 meters. And so the tidal channels are 10 meters deep, and you get down in one and you work in down there and get some fish with a net. And then you wonder how you're ever going to climb out in this mud. And so this is one of the muddiest places you'll ever be. But what I found is that this Galichthys that I was catching down there didn't look anything like the Galichthys that one typically finds across the region on the outer coast and in the Gulf. And so we collected them, of course, and did a phylogeny of them and did some dating. And what we found is that they are sister taxon to Galichthys mirabilis, and that they are about as old as the delta itself, about 4 million years old or so from what we could determine. And this was interesting because these fish had been described as a distinct species in the late 19th century, and very short, about a decade afterward, were synonymized. So what we did was actually resurrect this taxon. It isn't actually, this is not a biblical reference, but we resurrected it from synonymy. So it is now went back to being a real species. And so it is endemic to the delta. These are the sites that it's known from. This is the type locality from the late 19th century, and these are the sites that I've collected it at. And so it's in the delta, and it's also not very close to the delta. We find other sites that actually have Galichthys mirabilis, the other species that has this more mottled. And those estuaries or lagoons are different. They have less deep marsh, more typical salt marsh top habitats. But this whole area has been subject to water loss because we've taken all the water by damming the entire Colorado. So basically no fresh water gets to the sea in this region. And so guess what? It's saltier. So so this has some implications. And I'm going to talk about those implications more in the context of this genus of fishes, Colpicthes, and which was worked on by my master's student, Clive Lau. And so these two fish are both, this genus is endemic to the Gulf, but one of the species is endemic to the Delta and the other is more widespread in other estuaries. And so this is an interesting situation. And what we did is look at museum collections, not a huge number available, but we found that there was no evidence of historically of genetic intermediates or morphologic intermediates between these two entities. But when we measure the morphology from the large number of collections that I collected with others in this region, we found this basically place on the left-hand side of the Delta where there were hybrids, or excuse me, where we inferred there might be hybrids from the intermediate morphology. So all of a sudden, the distinction of these two species is coming into some question at the edge of the Delta. So we looked at a bunch of morphology, and then we looked at mitochondrial sequence. And what we found is there were some mitochondrial sequences from the widespread Gulf form in the Delta, both on the Delta edge and a couple of sequences from up in the Delta. And we then found some microsatellite loci that could work on these in the literature. And we found again that there was some evidence of hybridization in this on this Delta edge. And then we looked at, or Clive looked at some sequencing of nuclear sequences where we were able to identify hybrids, that is, excuse me, or identify heterozygotes that we presumed were hybrids. And so we found a number of situations where we could identify these hybrids in RAG1, a nuclear gene. And again, those tended to be concentrated at this Delta edge, but we're not all exclusive to it. So just to summarize these data, if we have genotype as represented by the microsatellites and phenotype as represented by the morphology using this approach here, we can see that we have these two major groups, the species in the Delta, the Delta endemic, and the species, more widespread species from all around the Gulf. And what we also find is that there's a number of hybrids that are, or heterozygotes from the Delta edge, and those are intermediate in morphology and genotype otherwise. And there's a couple intermediates that are not heterozygous, and then there's a couple of heterozygotes that are from up in the Delta and are morphologically like the Delta form or the Delta species, which is Colpicthes hubzai. So we see that there's hybridization. We also have this implication that there's integration, perhaps, of these Colpicthes regis widespread taxon genes into the Delta endemic. And so the concern here is that actually you're going to lose this species through the loss of the differentiation of habitat. Now that you've turned the fresh water off, you don't have the ecological distinctions that made these things isolated perhaps in the first place, and you're beginning to lose the distinction as introgression apparently is proceeding. So we're hoping to look at this with more with more transcriptomic sequencing and other kinds of things that will allow us to look at a little more at the genes that might be involved in this. All right, let's switch themes now from looking at extinct things, be they ducks or rivers, and worry about connectivity on the coast. And so if you were, some of you may actually be in New Jersey where the ice sheet advanced down to New Jersey. And so the whole Northern Hemisphere had much more ice in it on it, say 20,000 years and through a succession of glacial cycles. And when the ice was, when there's this much ice, sea level was lower. So the California coast, for example, had much lower sea level. Everywhere had much lower sea level when you had this much water tied up in glaciers. And so we can begin to think glaciers are known to have lots of effects on and form refuges for terrestrial populations, maybe some coastal populations where ice meets the sea. But we're pointing out that, in fact, these kinds of connectivity issues pertain through all kinds of other coastal habitats because of the glacial driving of sea level. And so we worked, on the West Coast. Here I am. I always wear this floppy hat in the field, collecting in Baja California. And we've collected lots of things in lots of places throughout this region. And so we have a lot of different samples, and we can begin to ask questions. And one of the things that I'll start with is the point that I'd like to make in using these data. We often think of marine species as very motile or where there's very good dispersal. And so there's very limited ability for differentiation and speciation. And so what I'm going to argue here is that gobies, which there are multiple gobies living in estuaries and lagoons on the Pacific coast, are more interesting than warblers because the way they partition habitat has an evolutionary effect. We love to think of warblers in terms of competition and how they've partitioned niches, but the niche partitioning that goes on in estuaries and lagoons of the Pacific coast by gobies affects their dispersal, because some of them are stuck in settings where dispersal is very limited. Others are in settings where their larvae can be dispersive very widely because they're have open access to the sea. So here's a confined situation, typical California stream-mouth lagoon, where you have a perched up body of usually very fresh water that persists through the summer unless it dries out. And then it's isolated from the sea by a berm. So there's many, many habitats like this. There's also lots of habitat that's confined in some intermediate fashion in this sort of context because, water's perched or pooled through tidal cycles up in various ways, or vegetation limits dispersal, etc. And then there are unconfined habitats where flow goes out in and out over broad flats, and the fishes, the larvae of the fish has immediate access to the open ocean and are presumptively more dispersive. And if we start looking at these gobies, here's a ellipnus. This taxon, the cheek spot goby. If you start looking at mitochondrial sequence, you find that fairly closely related habitats are a long ways apart. Maybe there's some subtle differences between the Gulf and the outer coast, but you don't have differentiation even over many hundreds of kilometers in open settings. Here's a taxon of considerable interest to various parties. We already talked about Galictes detrusis. This is within its sister Mirabilis, and we can see that there's lots of differentiation that's regional and local throughout the situation. And we can also see that in other gobies that are similarly have kinds of habitat that are in, have these qualities of being intermediate. And so these are the ones that are sort of interesting to explain, and they involve large, in this context with glacial cycles, because they are in these large estuarine habitats. Those are distinct from the tidewater goby, which is the most a locally differentiated vertebrate on the Pacific coast, and we'll get back to that in a few minutes. So, when we're thinking about sea level fluctuation and habitat, the sea level fluctuation through time interacts with the topography. Unlike, say, the East Coast of the US, there's a lot of topography on the West Coast because this is a very tectonically active area. So there's sets of steep coasts on here in these regions that are in barred form, where things drop off, especially at the edge of the, when you get to the edge of the shelf, they drop off very precipitously. And it turns out if you lower sea level, you get a very steep coast. You might have a little bit of shallowly dipping places even along this coast today, but at glacial low stand, when sea level was over 120 meters lower, these were very steep coasts indeed. And so the idea then is that you can't have estuaries in those contexts at low stand. And so we can begin to model that. So we're talking mainly about this last glacial sea level rise from about 20,000 years ago to today. And that's what we're concerned with, is what's going on here. So steepness matters then for how estuaries have been distributed through time relative to changing sea level. And if we do some examination of the slope, we can look at where modern estuaries form. And we can see that basically along the coasts that are marked in red here, those areas where the coast would have been at low stand are too steep for estuaries. And so, for instance, in this area in Southern California, northern Baja, we have 1 and 2. We have an area just north of Point Conception and an area north of Puerto Eugenia in the Vizcaino region. Those are the only 2 places where you seem to have persistent estuarine habitat. And that's sort of shown here. That is in green, we have where you could have estuaries at various different times as sea level rises from 20,000 years ago. These areas have continuous habitat, but all the other areas had a time period where there could not have been larger estuaries forming in the past. And so you have these refuges at low stand for this habitat type. And so we can start to look at taxa that are of the sort of kind that are intermediate in their dispersal potential. And here's microsatellite data from 3 such taxa. And we can find that there's potentially if we make a model of the Pacific coast, we come up with some ideas that there are 3 settings here that supported refugial populations. And then we can go and test those arguments using samples from a variety of places and putting various different models into a Bayesian computation and come up with some model testing to show that these data are not as well explained by other kinds of processes like isolation by distance or whatever. And so this sort of, in this case, these 3 habitat models or these 3 refuges are implicit and seem to be able to explain the data better than other taxa. So just to give Greer Dolby, who did a lot of this work in the lab, a plug. She has a number of papers out now that sort of expand on this theme, you know, from locally or regionally to globally in the relative to sea level and isolation of populations of coastal, especially estuarine taxa. All right, lastly, I just want to give you turn back to the tidewater goby. As we said, this is the most locally differentiated vertebrate on the Pacific coast. There's a number of management units that have been defined since this is an endangered species, and they're based on basically phylogenies we did a long time ago. And so I'm going to talk now about some processes going on in this area right here, just north of Morro Bay and near Hearst Castle, if any of you have been there on the Pacific coast. And so what we find in this region are rocky headlands that don't support habitat and 2 regions effectively that do support a set of lagoons, small lagoons. These are lagoons that then support tidewater goby habitat. And so one of the questions is how and when do tidewater gobies disperse? And the first observations we find is that they don't disperse across the rock, they only disperse across these sandy regions. And so we can see from these 2 time periods, instead of 1990 samples instead of 2008 samples, we can see that there's a rather isolating feature associated with that big rocky entity. And if we look at some assignment tests that were done with the microsatellite data, we can find that in 1990, there's no movement in this region. Whereas in 2008, in the samples we did in February of 2008, that there was a fair amount of movement. These are based on 30 fish each. And a few fish in many cases are found not to have been at their natal site. At least that's the implication from looking at all the microsatellite data. And so there's some movement. And so what's going on here? Well, in 1990, the presumption is that these lagoons stayed closed for long periods of time. And because there was not a history of rainfall from rain gauges and other data. But when it rains, these lagoons breach to the sea dramatically, and they then allow the tidewater gobies potentially to migrate. And here we are having migration of adults, seemingly only over sandy substrate. So this explains a lot about why we don't have them moving across rocky substrate, because they're not in the plankton. They're actually dispersing as benthic adults when all the lagoons are opening after major rainfall events, which was the case in 2008 when we collected right after a very rainy January. So this variation in rainfall is critical to a lot of what I've been talking about. So as I said, since I've been to California, it's rained a lot some years and not in others. And so you can imagine that there's a huge range of processes going on in these lagoons. If it doesn't rain at all, eventually the lagoons will dry out and fill in. If it rains a real lot, that actually restores the lagoons by digging them out. So there's a very dynamic process. These fish are living in this dynamic landscape. They are actually endangered. So we need to understand their metapopulation processes. That's why we're doing much of this exercise, to better understand what's going on, and to use these kinds of inferences about climate and breaching to make better models, better metapopulation viability models, so we can better preserve this listed species. And on that note, I'm going to stop talking. Thank you for listening. So if there are any questions about these topics, I'm happy to entertain you.
Thank you, David. That's a great talk. I'll start the question. I had a really quick question. So do you think the hybridization might be an issue for the other Delta endemics?
Yes. So the other Delta endemics include this Galictes detrusis. There's another fiddler crab that appears endemic. And so, yeah, so we will be hoping, or maybe, yeah, so we are curious if there might be introgression into detrusis from Galictes mirabilis. It certainly is a possibility. We haven't found significant evidence of that to date morphologically, but it seems like it is something that we're hoping to follow up on.
Great, thanks. Rene, do you have a question? Did I see a hand on yours? Okay. Let's see. If anybody has a question, you can just let us know. Or at this point, I don't see any hands right now, so you can probably just unmute yourself. I don't see any more questions. So it's 11:55, so it might be a good time to start the meeting. I want to thank all of you guys. First of all, I want to thank David for accepting the invitation and giving this great talk. Thanks, David. Thanks all of you guys for coming, and I hope to see you guys again. We had 3 more seminars, and if you're interested on the Twitter conference, just check out our website. All the links, if you just click on the Temporal Genomics Working Group link, every other link will be in there as well. So thank you guys for coming. I hope to see you again next time.